Before I start hopping into developing of new medicines in the age of AI and personalized medicine, just a question to the audience. Who here has some kind of life science background? Maybe raise your hands. Okay, maybe about 10%. So, sorry for you guys. I will not go too much into the very sophisticated biology and pharmaceutical background. I keep a little bit more on a high level to show you the concepts and the overall developments. So, the discussion about what drugs are doing, what's the importance of pharmaceuticals is not new. That has been on the horizon for many decades. It started in the mid-1850s, where you could buy at pharmacies opium and all other kind of stuff, and people started to discuss what's the meaning or what's the importance of drugs. And a lot of things changed since then. In order to understand that and put it into perspective of how AI is implementing some of these changes, we have to understand how drugs are currently made. For that, let's have a look on how the current drug discovery pipeline actually looks like. So this is what you'll also find in all kinds of textbooks, and I guide you through different items. So the very basic, or the very basis of research is what you've done in academia. So basically, zillions of PhD students and postdocs, and I guess some professors as well, contributed to a certain foundation that is the knowledge that we have. On top of that, we're going to start drug discovery. So, for instance, if you want to cure a liver disease, you figure out, okay, there's maybe a certain protein that's associated with the disease, I want to tackle that. There's a kidney disease, where there's a certain gene involved, I'd like to tackle this. So after identify potential targets, you start the validation of the target, because you want to make sure that what is written in academia, or in some papers, is actually true. So you run a bunch of experiments in test tubes, in petri dishes. You take a number of test compounds, add them on your cells, and you want to see if there's a certain effect elicited in the cell culture you're looking at, and hopefully this is something you want to see. If that's going well, and you have some starting compounds to deal with, you're going to optimize them. This is the lead optimization phase. Chemists are going ahead and modifying the compounds, making them more potent, more selective, essentially increasing the power to making them as a precursor of a drug. After that is successful, you go to the next step. You want to test the compound in a more complex biological system, laboratory animals. So you want to first see, can I reproduce the effect, and are there any severe side effects? If there is a severe side effect, this molecule dies in the progression path. If it works well, you move to the next phase. Next phase is the human. You usually start with healthy human beings. You want to first make sure whether the drug is toxic, there are severe side effects. If that's not the case, you go into patients, and you want to see whether the whole proof of concept actually works. At the next step, you're going to a larger population, and you want to see whether there's a therapeutic effect which is beneficial. If that all works out well, then you ask the regulatory authorities for approval to get your drug on the market. In US, it's the Federal the Food and Drug Administration. In Europe, is the European Medicines Agency, and they evaluate the data that you have. They ask for additional data, ask for additional studies, but at the end of the day, you're successful. You get the approval to put your drug on the market. That means you, on one hand, get rich. On the other hand, the so-called phase 4 starts. It's a long-term study. The drug is on the market, it's prescribed by doctors, and then there's a continuous surveillance how the drug actually works. So that could be that a doctor describes a drug to a certain patient who has a, heart condition, and it turns out, okay, this drug doesn't work well, there's severe side effects. That means for future prescription, this drug is refined. Everyone who has this condition is not getting the drug. This happens after the drug is on the market. So this is essentially the whole drug discovery pipeline in a nutshell. You can appreciate this is a pretty complicated and complex issue, and it takes a lot of time. So the whole drug discovery part is maybe 3 to 5 years, preclinical development 1 to 2 years, clinical development 5 to 7 years. The authorities take another 1 or 2 years. That are all average numbers. For some, it takes much longer. For some, it's much faster. But if you look at a textbook, this is roughly the ballpark you're looking at. The rate of success is also pretty hefty. So in average, you start with about 10,000 compounds at the very beginning, and only one of those makes it to the market. Altogether, we look at an average drug development time of 11 to 15 years, so 13 average, and the costs are currently more than $2 billion. So this is a really long commitment, much of time, and a lot of money. Why? Why is this so risky and why is it so expensive? So there are several reasons to it. One reason, or in general, the main reason is the so-called translational gap, or also the valley of death. It sits in between the body of knowledge from academia and the application in the human being. So something in between goes wrong, and this is the reason for the high price tag, and also the reason why about 90% of all drugs will never get on the market, will fail in between. The reason for this valley of death, there are several. One of them is the problem of academic knowledge in general, and I think we had to talk about this several years ago here at the Congress as well, the replication crisis. The body of knowledge we're dealing with is maybe not as robust as we hope it to be. There are some examples in the field of cancer research. There's the Open Science Center for Open Science, who started a reproducibility study looking on how reproducible is the knowledge in academia if it comes to cancer. So they tried to reproduce 200 experiments from about 50 papers. A third of them, they could not even start it because the information wasn't there. About half of them they could start, but they were not able to complete it. And the last quarter, they actually could complete, but maybe only by their metrics, half of them were successful. And usually the positive effects described in literature were not as strong as they could reproduce. So this is already an issue, and if you want to spend $2 billion on this basis, that's maybe a little risky endeavor. And the pharmaceutical industry is aware of that. So there are a couple of studies. The first one from Bayer is from 2011. They also looked in cancer studies and found that maybe only a quarter of them they could reproduce. So similar ballpark numbers as the Open Science and the Center for Open Science described. Amgen, another company, did something similar. They were only able to reproduce 11% of all the academic literature. Interestingly, what I found in this study from Amgen, they put in this table, which you can see here. And even if there are studies where experiments are presented and they could not be reproduced, those are cited heavily and frequently, creating a large trail of other papers referencing experiments you never can reproduce. So that is a growing number of data of papers on data, which is maybe a very weak foundation for any kind of scientific endeavor. So if you take all the public literature, put it into LLM, and ask ChatGPT, hey, is this maybe a good drug? You run into issue because you have all these biases here. The LLM is not able to reproduce the experiments and is not validating and score them, whether this is a good thing or a bad thing. So this is one issue on the scientific foundation. The other issues are centralized around the technical and experimental setup that we have in drug discovery. So this is another representation of the pipeline. And what you do in experimental research is you want to figure out whether the biology does what you hope to do, what you hope it does. So you work with standardized cell lines that have been extracted from patients decades ago. They're cancer cell lines. How much do they represent the real human being? Maybe not so much. We all know that, but it's one of the models that we have. The other issues are animal models. They are standardized lab animals, inbred for decades, used as a model to replicate a human disease. Maybe there's some limitations to that. Again, everyone knows it, but there's only so much you can do. Drug delivery to animals works very differently. You can inject drugs into the bloodstream of a mouse, and it goes into the brain, which does not work in humans because the brain is shielded differently. So there's some issues there. You also can administer drug in the mouse in a very different way. You can inject it into retroorbital veins, so right behind the eye. In theory, you could do it with humans as well, but it's maybe not applicable. Historically, we have a lot of data on clinical trials, but they are usually including male Caucasians, so there is a lack of representation of women and minorities. It's getting better, but historically it's still there. The other issue, if you look in a clinical trial, you want to see whether something works, you focus on symptoms. You don't necessarily understand what happens on the genetic, on a molecular mechanism, so you might miss something. You're flying blind to some extent. And this is also bleeding into the selection of patients. Let's imagine you want to put a drug on the market against a liver disease, and you have patients who have liver disease A and B and C, but all look the same, we all have the same symptoms, and you all give them the drug, and the drug only works against liver disease A. People with B and C, they will not respond. The statistics of the clinical trial look bad. The trial fails, even though if you could only select A, exclude B and C, it would have worked. So this is one issue that we have in the drug discovery world. How can we make it better? And what is the role AI can actually have here? So, as I said, there are a lot of issues. AI cannot fix the issue of the knowledge base that we have, but it maybe can help us here accelerate the process. And without going into too much detail, there's a lot of work in finding better in vitro models, better mouse models, having better drug delivery systems, recruiting more minorities and women into clinical trials, getting more data to understand what happens in a patient, and using this to also select patients more adequately to a clinical study. So this is all happening, but as you can appreciate, all these come with data, data, data. And this is the foundation for any successful AI model. Now, if you ask, I recently was in a symposium where that question was asked among the attendances from pharma and biotech and academia, where do you put your AI capabilities? How advanced are you? And basically this is a little bit what the audience said, and I think it's quite pragmatic. 2/3 of the audience said, yeah, we're scouting, testing, playing around, seeing how it works. Some of them said, it's part of our strategy. There are some applications, and only a very small number of attendants said, yeah, we see some beneficial usage. So given that, I think we have still some time before AI takes over, nothing to worry about. Though there are some applications that are currently integrated into current workflows, and I have here some examples. One example is AlphaFold, and I guess that has been presented, not the last one, but the Congress before, I think. It helps to predict the protein structure. So imagine I want to build a drug against a protein which I have no idea how it looks like. I can come up with hundreds of experiments I can do, but if I get a good estimate how a structure could look like, I can already start with 20 experiments first and put some other 80 on the back burners, and maybe the first 20 are already successful. So it helps prioritizing the experiments I can do, which is already a huge benefit. Image analysis is another thing. So if I take samples from a cancer patient for what's the tumor looking like, or if I take animal tissues to want to see whether there is an effect I hope to observe, I have to take the tissue, I have to prepare the tissue, I have to stain it, I have to put it under a microscope, get the right resolution, and then count the different patterns I see there. This is a lot of laborious work. In the past, it has been done by someone looking in a microscope and checking boxes and counting light dots. Tedious work, but over the years, more and more of this has been automated. You can automate image analysis, quality control, all of this. This takes away a lot of tedious, laborious work of someone sitting in front of a microscope. It can be automated away. So this is happening. The other thing on the right-hand side is how to optimize your compounds. There was an article from the Chemical Abstract Services, part of the American Chemical Society, who looked into a couple of patents or drugs that have been put out into human clinical trials, who originally have been created by AI. Their conclusion, or the conclusion of the author, was so-so. It's okay. It's not that exciting, but it's something that is happening. So we are early there, and already AI tools helping us to design and optimize small molecules, making them better, turning them to potential drugs. So conclusion of what's happening here right now is, yes, there are AI applications. They're already part of the current workflows. They help prioritizing experiments and timelines. They increase efficiency, and they increase productivity. That's pretty good. So far, there has been no impact on the human workforce. They're still there because, especially if it's R&D, you have thousands of questions, and if the AI helps you to answer 5, there's still so much more you can do in the other time you have. So that's ongoing. That being said, there's one recent example from a company, I think it's in silico medicine, who have first identified a target for lung disease and designed the molecule using AI from the very beginning to the very end. So essentially, we have been able to start from this part and going all the way to the first part here within 2.5 years, about 30 months, which is, in average, 3 times as fast as conventional methods happening. And they've been even able to complete, August last year, the first part of the clinical phase 2. So we're quite advanced in this development pipeline with rather a little bit amount of time. Now you can argue, well, there may be hundreds of companies working on AI, 99 fail, and just one got lucky. They could be the lucky ones, maybe. There could be also, the other thing is, these are average values or average timelines. For some drugs, it takes much longer. For some, it takes much faster. It's hard to really put this one case into this pipeline. Everyone is looking at it because this is kind of the prime example of how drug discovery can be accelerated by AI, and this is kind of a little bit the pilot. Everyone is looking what's happening. So there might be something happening, but I think it's a little premature to come to a conclusion here. Whatever you do, if it comes to AI, you need a good, solid basis of data. So good data is key, and I think it's nothing new I'm telling here. I think you all know that. So good data is, and then there's a series of different adjectives you can put on: accurate, comprehensive, curated, reliable, and so on and so on. I think you get the gist. If it comes to data from a biomedical context, it's a little bit tricky because you have to obtain the data from living organisms, from patients, sometimes across different countries, across different clinical centers where different people work. In order to get data that you then compare among different patients and countries, you have to make sure that they are all adhering to a detailed standardized protocol. You need standard operating procedures. Data needs to be curated. You have to make sure that the people handling the cells, the samples, the biological specimen, adhering to a technical and experimental level which matches a certain accuracy level. You have to be able to trace everything that's happened. So this is really expensive, time-consuming, and costly. Small academic labs or small clinical institutions are not able to have the manpower and the resources to do so. This falls back to larger companies and or specialized companies, or in some cases, large national initiatives like the UK Biobank, a large nationwide cohort study where they've actually pulled the resources together to gather that data. Those data is costly to get, and if there's data banks, they won't have access fee or you don't get it at all, so we're limited access to the available data. What we also see is a lot of large pharmaceutical companies have been on drug discovery markets for decades. They have internal data sets from R&D projects 20 years ago, 30 years ago. All this data they have usually obtained in an industry setting, so it's higher standard, higher traceability versus an academic lab. They have clinical data, and not everything they have submitted to the authorities. Or you don't have to, but they are pushing to get more data to be shared, but in the past it hasn't been the case, and especially clinical trials that failed. They never submitted those, but they have the data and can learn from it. So they have this body of data, plus they have their own cohorts or bought into cohorts and know a lot about different patient groups. So they have a lot of data in-house, and this is kind of their proprietary data sets, which are high-value assets. And this is what they use to put AI on it, to come up with new ways to find a new drug and drive forward the drug discovery pathway. So if there is anything value in data, usually what happens is some people figure out, oh, there can be some money with that. And this is an issue in the US, but it's also an issue in Germany and Europe, that cyber incidents happens all the time. If it hits a big pharma company, it can be really expensive. But if you're a big pharma company, don't worry, you're not alone. It happens all the time. We also saw during the corona pandemic, especially the frontrunners in the COVID-19 vaccine race, they have been targeted, but also others who were involved. And as I said, clinical data are very important. They should be protected from cyber criminals, but I guess also from users, because that's usually a problem. Cybersecurity is not a new trend. It's becoming more important in the drug discovery and pharma world as well. But there's one particular discovery or trend in drug discovery I'd like to focus your attention to. And this is the shift towards personalized or precision medicine. The overall concept is, if there's a drug, if you have high blood pressure, for instance, you get a certain drug and then hopefully it works. It could be, and maybe you know it from people you know, from families or friends, that a drug works for someone it does not, someone gets a headache or doesn't feel right. Some people respond differently to drugs, and if you would know that, you could say, okay, this group A, you maybe get this drug, and group A, you get maybe half a dose, and group C, probably you don't take this, take something else. That would be great, because it would make the drug treatment more efficient, and it would reduce the amount of side effects that you have. If you go one step further, you end up with personalized medicine. So you take the consideration that we have all a unique genetic makeup, because as you know, we are all individuals, and everyone has to be treated in a special way to obtain the largest effect possible. But that comes with, well, pros and cons. The good thing is the promise and the idea of personalized medicine, instead of have one-size-fits-for-all medicine that you can give to everyone, and some are benefiting from the effect, some have nothing, and some actually have side effects, you would have the idea that there are some drugs that work for person A, another one for person B, another one for person C, and they all would get the best benefit. So this is the idea and the promise and the hope of personalized medicine, which I think is quite good for the patient because you get the best treatment possible. The downside comes right here if you push it even further. Pushing even further, I specifically want to refer to genetic medicine or genetic therapy. There are genetic therapies out there. I put here 4 of the contemporary examples. They're all targeting rare diseases, some very, very special diseases where no cure available. From all kinds of different companies, and it's usually one therapy. You undergo one therapy, but this one comes with a huge price tag, between 2 and 4 million, depending on what it is. So they're expensive. For people who are suffering from this disease, there is no treatment, maybe except that one. They suffer and usually die in a very young age. So for them, gene therapy is a big thing. It changes their life. It actually saves their life. However, strings attached are several. On the one hand, the one on top, Lenmeldi in the US, in Europe it's called Libmeldi. The company said in 2023 they generated about $12.7 million with that. If you look at the numbers, that's maybe 3 patients. And in 2024 it's 22.2 million. That's maybe 6-7 patients that are super small target populations because it's a rare disease. It doesn't happen often in the population. There are only few people who have it. For them, this disease makes a difference. This treatment makes a difference. But this treatment is not trivial. It's really, really complicated. You need specialized centers, you need specialized doctors, you need specialized infrastructure, you need specialized care. That's one reason why it's so expensive. It can't be done everywhere. You have to really go to specialized centers. And with the cost comes the question, who is covering the costs? And this is a big issue. If you're in a single payer environment, maybe. In the private insurance, you're probably screwed. And there comes also automatically the ethical question. This question has been around forever since there was the idea of gene therapy, but it's becoming more urgent because we already see gene therapies happening. So what disease should we treat? Who decides that? And at the end, who gets it? Is it a decision or an agreement that society makes, or is it what money allows you to do? So this is a question where no answer is there yet, but it should be answered sooner or later because we see gene therapies and there will be more of them, and these questions are important to answer. There's one more development which I'd like to focus your attention to. And I called the end of the power cycle because I think it fits nicely to the overall theme of this Congress. We have here this drug development pipeline. This is what you've seen. It's expensive, $2 billion on average, and it takes a long time. For a pharmaceutical company, you need some kind of revenue idea to fund this drug discovery pipeline. And this is the concept of blockbusters, which coming in. Blockbusters, you know me from movies. It's this one summer movie where everyone goes there and it earns a lot of money. The same thing applies to drugs. A blockbuster drug is a drug that formally exceeds $1 billion in annual revenue. If you adjust it for inflation, it's maybe more like $3 billion. And here are the top 10 from last year. They make about $10 to $30 billion for Stelara with 10 and Keytruda for about $30 billion last year. So there are the essential parts for pharmaceutical industry to actually fund and drive forward the research and development pipeline. Without these guys making the money, it won't work. Why is it possible to earn that amount of money? So there are several reasons to it. One reason is patent protection, and I picked the 2 recent examples of Ozempic and Wegovy. Ozempic, if I go back, is also number 3. It's not long ago that it came to the market, but it's already having a huge effect. So Ozempic is a treatment for type 2 diabetes. It's an injectable pen, and the compound that actually does the magic is semaglutide. In the US, you can get it for $1,000, in Germany for maybe 60. I'm not going into discussion why we have discrepancy. That's a whole different story, which I'm not starting here to address, because I have to stop in maybe 10 minutes or so. The other part is Wegovy, which is basically the treatment for obesity. It's an injectable pen with, surprise, surprise, semaglutide. It's slightly higher concentrated, but other than that, it's the same as Ozempic, and it costs more because you treat now something different. Why is this? Different discussion. There was a study last year who they tried to figure out, so how much does it actually cost? What is the production cost essentially? And the group, they came to a price tag between $1 and $5 for that. So you already see there's a pretty significant profit margin embedded into the different prices, if I put it carefully. This is one of the reasons for blockbuster drugs. You have high demand and you have a high profit margin. That only works because we have patent protection. If that ends, we come to this. And this is an example from a couple of years ago. It was Pfizer's Lipitor, a drug to reduce cholesterol levels, if I'm not mistaken. It was bring in a lot of revenue, between 9 and 13 billion per year, until 2012. The patent expires, and they usually do that after 20 years, and if there's a huge drop of about 80% within a short amount of time. It means that money is gone, because now the exclusivity is gone. Other companies can bring on the same drug on the market and maybe ask for half the price. And you've seen the profit margins. They can even go to a quarter of the price and still making profit. And this is what happens, and this is the reason for this huge drop in revenue. For a company who has 1 or 2 blockbusters, this is a big issue because they would suddenly lose all the revenue. Now, what's happening right now is this patent cliff, as they call it, is coming, and it's coming big. More than 20 blockbuster drugs will lose the exclusivity within the next 4 years. More than 20. That means we have one of the largest, even the largest patent cliff ever in the pharmaceutical history, and about $230 billion billions in annual sales are at stake. If you don't know what number is, it's maybe 20% of the entire global prescription market, or in Germany, it's 1 Doppelbums. So this money is about to kind of vanish or move into other companies, which are not the pharmaceutical companies, because there's no patent protection anymore. With that developments, we see some interesting situation in the whole pharmaceutical landscape. And if I summarize that, we have on the one hand this patent cliff. The revenue model is in jeopardy. We have more generic drugs, which can already treat a lot of disease. So if you come up with a new drug, you can't really ask for big premium prices if there's already something else. It has to be really, really good compared to what is on the market. Otherwise, you can't ask for a lot of money. So that really put a lot of pressure on the drug development pipeline and the people doing the research and the companies. Then we have precision and personalized medicines. Patient population is getting smaller. Treatments become more complicated. It becomes decentralized. How is this working in the whole concept of having a blockbuster? It doesn't really work. On the other hand, large pharmaceutical companies, as I said, they have this huge amount of data which we collected over decades. And they have it. It's usually on a pretty good shape. We have a lot of information. And the hope is that AI can be used on this data to identify ways to reduce costs, to accelerate the timelines of drug discovery, and eventually also to identify new medicines with new benefits for which you can ask a higher price. If I take all that together, we always think, oh, AI is disrupting the industry. Here, in that particular case, I don't think it's the case. It's more the opposite. It's a necessary development. Otherwise, it wouldn't work. And I'm personally not really worried about AI in that field. I'm more worried about the patent cliff and the shift to precision medicine. This is very, very good for the patients because you can address a lot of diseases you cannot address before. You can improve care substantially. The question is, to what condition? What defines the accessibility to a certain treatment? Is it general societal consent, or is it money that decides decisions? So that are big questions. Risk and uncertainties come here close to the whole promise of precision medicine. I think that's something we should discuss. And I wanted to kind of summarize the current developments, give us some food for thought, and yeah, that's all what I had. Thank you very much, and happy to take questions.
Thank you very, very much, Dennis, for your talk. And I think there will be a lot of questions. Please go to the microphones in the hall. Please, at the microphones in the hall. Yeah, at one.
Hey, yeah, thank you. Super interesting, this whole pipeline. Can you maybe give some examples? I mean, what is typical data that goes into these AI models, and what is the output of these AI models? So is it like studies go in and the protein comes out, or yeah, just maybe 1, 2 examples how this actually works?
Everything and nothing. It really depends on what you're looking at. For instance, I don't want to go back all the way, but you remember some of the images where you analyze microscope data? Then it's a very limited data set. You use a specific microscope data. So it's for that particular case, it's a narrow data set. It's very limited. If you go for larger drug discovery, where you want to figure out on what could be new target, then you're going more broad. You're going to take consideration all academic literature. You go for patent applications. You're going for grant applications. All clinical data you can find. Either they're publicly available, or you can find them from competitor information, from competitor companies, internal data. All that goes in, basically everything you can get. The idea is the more you have, the more you can narrow down some things and exclude some other things. The issue is usually accessibility, because it's really hard to get comprehensive and highly curated data. They are really precious. But that's essentially in a nutshell. That answers the question?
Yeah, thank you.
Perfect.
I think we have a question from the internet. Signal Angel, please.
Yes. Can we also use LLMs to improve the tests that doctors give to patients to determine what drugs to prescribe?
I mean, you can use everything. Yes, with a big question mark. I think we are not there yet. It's maybe a little bit outside of my really expertise what's happening on the clinical application. If you want to understand the disease or general symptoms of a disease, it's good to take a lot of things in context. That's difficult for a doctor. But I guess there's also the idea that if you get a lot of data from the personal patient history, plus some genetic information, some drug treatments in the past and the present, with all information compared to certain databases, and an LLM can point you in some kind of direction. I think the idea is there, that it could be a benefit, but I don't think we are there yet. So maybe ask me the question in 10 years again, and then we'll see.
Thanks for the question, and thanks for the answer. The next question from the microphone 6, please.
Yes. Do you think the use of AI will enable small and mid-sized companies to get into the pharmaceutical market more?
Yes. Yes, definitely. Now I have to go all the way back. Sorry about that. This one example I just showed you earlier, that one. I think in silico medicine is a rather small company. And they use the AI model that they have to really come up with a novel target and move it all the way to the human trials, phase 2. That's something that can actually happen, and we see that for some smaller companies that it actually is happening. The other thing is, the question is how long do they remain small? Because if there is a smaller company, you have something very interesting, like a good drug or a good AI model, big companies come and usually buy them up. That happens. But generally, if that accelerates the timeline, if you cut down the resources, if you have really good AI model for something very specialized, yes, this is really an open door for smaller companies, and this is what we see. Yeah.
Thank you.
Thanks for the answer. The next question comes from the microphone 4.
So thanks for the talk. It was very interesting. So one thing you mentioned that was that medical data sets have a huge bias for like white male Caucasian people, right? And That's like a known fact. And then it's also known that AI sometimes even enhances these biases and not lessens it. So how can we counteract that, basically?
That's a good question. I'm not sure what the answer is. Especially historical data has this bias, and there's nothing you can do about that unless you kind of fix AI, which I don't know how it should work. But we see more modern clinical trials where they have a different recruitment scheme, and since it's more modern clinical trials, you have modern technology, getting more data there. We have a little bit the discrepancy between old clinical trials with smaller amount of data and larger clinical trials of modern ages of larger data. So there is maybe the chance that over a couple of years, the more modern clinical trials who have more data will outrun the old, and then we have kind of limited the bias. But it's something that will take some time, and for now we have to be aware that a lot of these things have the issue, as you just mentioned, and not only for the human diversity issues, also everything in terms of biology. We use certain cell models, which are still old. We use old animal models, which not really reflect everything. So there's a lot of bias, not only on the human level, but also on the experimental side. And this is something we have to address and keep in mind whenever we use AI or something like that.
Thank you. Microphone 5, please.
Thank you for the talk. What do you think about every cure? For example, they try to repurpose existing drugs which are approved for other diseases.
What's the name again of the company?
Every cure.
I'm not sure if I heard about them. But I think they are not the only one. I think that's a general idea that many companies try. Like the example of Ozempic on Wegovy goes in the same direction. It's the same compound, but it goes for 2 different indications or diseases. And for a drug development, that's kind of a good way because it can shorten the clinical trials because a lot of things you work already apply for a new one. So it kind of reduces the development pipeline and costs. So this is something they try to do. If that's their business model, yeah, I think we're not the only one. A lot of them do it. It's part of the entire business. Another thing is, if you want to develop a drug for a rare disease when no drug is available, you get some kind of beneficial drug discovery credentials from the authorities. Some things you can reduce, some smaller clinical trials you can run because there's no competition. And if you have then used this to get a drug on the market, you can use this to go for a different indication, and then it's already cheaper. So there's all different strategies, and it's rather standard in the industry, I would say.
We have another question from the internet.
What do you think about using AI to simulate entire cells and protein-to-protein interaction?
It's cool. I think it's a little bit too early because the AI is very nice and great, but it depends on the data, and the cell essentially is a black box. We know only a little bit. There's so many proteins which we don't even know how they look like. There's protein modification. There are all different kind of lipids floating around, and no one cares about lipids. There are other parts in the cells which we don't really take in consideration. I'm not sure how much of the real amount of unknown we actually can include into the current procedure. So modeling a cell on protein interaction is a very limited, narrow point of view of what really happens on the cell, and we don't only know a small portion of it. So it's a cool idea, but from the real application, I would take it with a grain of salt.
Thanks. We have another question from microphone 3.
Yep, thank you. I have a question concerning the reproducibility of studies you mentioned earlier. The problem is not fixed by AI. How can it be that so little can be reproduced? Do you have some insight on that?
Hang on. There are, I guess, a whole host of different reasons. One reason is we work with biological materials who have this annoying feature that there's so much happening which you don't know. If you keep cells growing for some time, they change and you might not know it. They change their metabolism, maybe there's a mutation occurring, and that already leads to to different outcome. You feed the cells with certain cell media. You hope it's the same, but you don't know. Could be some minor changes here. Something is different in the I don't know, air system, which is keeping your cells in a sterile environment. There are a lot of so many small things you can't really control. That's one element of contribution. Technical feasibility of a PhD student versus a seasoned technician makes a huge difference. And then there's, of course, in academia, the pressure to produce publications. So maybe you ignore a couple of effects and push out that. So you ignore a lot of negative data and only publish the positive data. So that also is some of the elements. So there are many, many reasons. That are some of them that just came up to my mind.
There is another question from microphone 1.
Yes, thank you a lot for your talk. I have a small question regarding your opinion on the possible effects of the AI studies on the clinical trials pipeline. Well, we all know why the clinical trials are so lengthy now. We had thalidomide and other issues which made people, put some safeguards and make the trials quite long, comprehensive. What's your opinion? Might we get more effective data treatment with AI? Might the clinical trials be more, compressed, more efficient?
Good question. I know there's some developments. For some antibody developments, I think the FDA put out you don't have to do animal trials anymore because we can reduce toxicology information, I believe, from already other previous studies and test tube experiments. That's one thing that happens. Some of that could probably bleed into human studies as well. One of the issues is personalized medicine. Groups of patients getting smaller and smaller. And maybe there's a disease where only globally 1,000 patients. You're not able to run a large pharmaceutical clinical three study with 10,000 people. It's just not possible. So you have to do something smaller. And for a smaller disease where no drugs available, you can get away with much smaller groups because this is a specialized case where the authorities and regulatory authorities know about that. So we will probably see more of this if you're able to subset your population based on precision medicine to smaller groups. That's maybe a development that we can see if they're getting smaller.
Thank you.
Okay, thanks. Maybe the last question from microphone number 4.
Yes. Hi. Very nice, actually, presentation. My question are, in fact, 2 questions. If there is, if there exists any effort or about develop a standardization or protocol of use AI in clinical trials to avoid based tools and how to perform a blind study using AI. Because basic results cannot come necessarily from basic data sets.
I'm sorry, I'm not sure if I got the question.
The question, is there any effort any effort to develop protocol or standardization to avoid basic results or to perform a blind study?
It's probably a little bit out of my comfort zone. I'm quite sure the authorities know about the rise of LLMs and AI and all those tools, and they take that into consideration. I think there are also active working groups and committees to think about how could this impact the study designs that you have, the requirements for clinical trials, how we can shape this. And if it comes to that, then of course I want to have a very standardized protocol. What kind of AI application have? What is the data set? What's the training set? What are the different steps you apply? We want to have the open source AI toolbox, for example, available. So these things are probably under discussion. I'm not aware that there's something out there. I might be totally wrong here, but I think it's in the making and the process are developing, if I'm not mistaken.
Thank you. Okay, thank you for your talk. I know there are a lot of questions, but we have no time for this. Where you can find you at the Congress?
I'm floating around, maybe somewhere outside.
Okay, thank you and a big applause for Dennis.